Hi guys, today we are going to be talking about mitosis. As you know, mitosis is the division of somatic cells. Here is a diagram that shows the stages of the cell cycle. Here is G1 where the cellular contents are duplicated. Here is the S phase where the chromosomes are duplicated. And here is G2 where the cell double checks for any error in the DNA. For example, if this phase wasn't here, then cancer would be much more common because mutations would be more common in the human genome when the cells replicate. But we will not be talking about these three stages because they are part of the interface, and instead we will be focusing more on mitosis and cytokinesis. Here is a brief summary of mitosis. As you know, mitosis has four stages: prophase, metaphase, anaphase, and telophase. Cytokinesis is not part of mitosis, but it happens right after telophase. Here is the first phase of mitosis, which is called prophase. Prophase is when the nuclear membrane disappears and the genetic material began to condense and become chromatid. Centrioles also began making spindle fibers. In the real diagram, you can see that the genetic material is visible and the nuclear membrane is disappearing. Metaphase is the second part, and it is when the genetic material lines up at the equator of the cell. Here in the real diagram, you can see that the genetic material is lined up in a single line at the equator. The third phase is called anaphase, and it is when sister chromatids are pulled in different directions. As you can see in the real cell, each genetic material is moving in different directions. And the last phase is called telophase, and it is when the nuclear membrane reforms and the genetic materials began to uncondense and become chromatin again. Also, the spindle fibers made by the centrioles get destroyed and disintegrate because they are no longer needed. And in the real diagram, you can see that the genetic material are at opposite sides of each other. As you already know, there are many differences between an animal cell and a plant cell. One of them is how they undergo mitosis. In an animal cell, as you can see on the left, there are centrioles and the daughter cells are separated by a cleavage furrow. And on the right, as you can see, plant cells don't use any centrioles, and the daughter cells are separated by a cell plate made out of cellulose. Here is a diagram. On the left, we have an animal cell, and on the right, we have a plant cell. For example, as you can see, during prophase in the animal cell, there is centrioles making the spindle fibers, and in the telophase part, there is a cleavage furrow forming and pinching of the cell. But in the plant cell, during prophase, you can't see any centrioles, and during telophase, there is a cell plate forming. So that's why plants in real life can't move, because they are always touching each other and they're only separated by a cell wall. Now you might be wondering why mitosis is important. Mitosis is important because it allows an organism to grow. For example, if your cells didn't undergo mitosis, you would be the same height as when you were a baby, and you wouldn't grow any higher or taller than you are right now. Mitosis is also important because it allows your body to repair damaged cells or tissue and repair wounds. It also replaces dead cells or tissue, and it also helps in reproduction in some simple organisms. For example, yeast cells replicate by using mitosis and not binary fusion, because they are not bacteria and they are eukaryotes. And I just wanted to thank you guys for watching.